Hello, everyone. I would like to welcome you all for today's free webinar, which is about a special opportunity to learn modern biotechnology and life science techniques using innovative Omicslogic research-based online training. My name is Sonalika Ray, and I want to start by introducing our company, vision, and team that stands behind the various resources and training programs we will go over today. Omicslogic is a program for bioinformatics training that was developed in collaboration between Pine Biotech and the Tauber Bioinformatics Research Center at the University of Haifa. The company was started as a result of technology transfer from the center, which was founded by Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Alfred Tauber, who comes from a medical background, has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center at the University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career, he has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team includes experts in bioinformatics, computer science, and training. Some of our team members will be presenting today as well. As a result, we have acquired professional certification to offer our curriculum and support research projects using our Omicslogic program project-based curriculum. Over the past several years, we had the privilege of working with a number of organizations developing a training program for research-oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. Using the cutting-edge Omicslogic resources, we bring a unique opportunity to learn computational biology and apply data science techniques by working with experts bioinformaticians. And in today's webinar, we will go through the introduction to the program, followed by an overview of basics of bioinformatics and big data, concepts and applications, genomic medicine and sequencing data analysis in biomedical research, application of microbiome and metagenomics analysis in life sciences, and transcriptomics data analysis and its application in the machine learning. Before we go through the topics that will be covered in the program, as a community manager, I interact with participants who are interested to know if they are eligible for the training program. Let me explain that. So this program is a great fit for students who are interested in learning about the ongoing life science research and to develop skill sets to analyze genetic and clinical data. This program is also a great fit for researchers who are keen to apply computational biology tools to their research projects and faculty who are working on the research projects and proposals and have a keen interest in teaching bioinformatics. The industry-driven Omicslogic curriculum helps biologists and clinicians working with industry and research labs to analyze their own data and take decisions based on the analysis. This program is available through our collaborators and mentors in the US, Canada, India, Sri Lanka, and for the students and researchers in Africa as well. Now to talk more about Omicslogic bioinformatics training and computational biology program, I would like to invite Dr. Mohit Mazumdar, who has a PhD in computational biology from Jawaharlal Nehru University and is an author of more than 30 international publications on bioinformatics research. Over to you, Dr. Mohit.
Thank you, Sonalika. Thank you, Sonalika, for the introduction. Hello, everyone. We are absolutely delighted to have you all today for this webinar on Computational Biology program. For everyone joining for the first time, my name is Mohit Mazumdar, and I am a bioinformatician by education. And since last 12 years, I've been practicing and applying bioinformatics for research, and now for both research, training, and collaborations. To all the participants who have joined us today, it would be really nice to know your background, educational background too. And I saw that many of you have already shared with us. It's so nice to know about your research interest, about your educational background, so that we can understand and guide you better. So, if you are from life science, then you must have studied biotechnology, right? So, biotechnology has been in use since 1919. So it's a long time back, and it has its first application was in DNA recombination experiment with bacteria, right? And today, biotechnology in itself has been applied in manufacturing of medicines, medicinal devices, diagnostic tools, biomaterials, food technology, and food production, all of that. But today we wanted to talk to you about another exciting field, which is computational biology. So, computational biology, which is a relatively newer field of research, has been evolving at an exponential pace. And it's such an exciting field of biology. And we will learn more about it in today's session. Uh, and to talk a little bit about computational biology, there's a reason that this exponential pace that it has got. It is because of the enormous amount of biological data that has accumulated over the years, and the size of this data is ever growing, and the application of biology in life in itself is growing already, and it has been shown even for the experiment that some scientists did a few months ago, that DNA can be used to store data just like our typical hard drives, right? Scientists were able to show that 700 terabytes of data could be stored in a single gram of DNA. So that's like pretty exciting, right? So, but our objective here is to simplify complex bioinformatics methods that have been applied in these amazing experiments. And then we want to use these methods effectively and efficiently to tackle unsolved problems in biology and in healthcare. So I would also like to tell you about in much more detail about the Omicslogic program. But before I do that, I would like to ask if any one of you would like to have a discussion, a question, please feel free to put it in the chat option so that we can go through this later and answer your questions. So going back to the presentation and about the Omicslogic programs and what we've been doing and been talking about. So the goal of Omicslogic program is to provide training in bioinformatics, enabling independent research guided by mentors and peer examples. Another aspect of important aspect of Omicslogic program is to develop a growing community with a shared passion for data-driven research and application of citizen scientists science projects. So we have a growing community of over 10,000 people who are computational biologists, researchers, biologists, clinicians who interact with each other and enrich this information sharing and collaboration. The third objective is to enable students and clinicians and faculty of all background, people who are interested genuinely would want to learn more about it, apply these skill sets in their research areas for job opportunities, and to develop noble and independent research using life science or the latest technologies in bioinformatics and computational biology. So these are some of the objectives that we wanted to tell you all about. And the next thing is about the program itself, because we would want to complete some of these objectives by going through a training method. So this online program is designed for everyone, including students who don't have a background in bioinformatics. So the topics covered in this program enables anyone to learn the NGS data analysis, because we will be going through different kind of processing sections but for doing different sort of analysis, such as genomic analysis. And I've heard many of you are interested in transcriptomic analysis and interpreting the omics data using machine learning techniques. So we will learn about along with this, we will also be learning about public repositories where various file formats are being deposited by different laboratories and companies, and their application, and then understanding their application for biological research. So this analysis will include, we will be doing annotation, visualization, and interpretation of the results. So before we go into those technology, let me start today by mentioning that as a company in this space, we are focused on addressing some of the pressing needs in biotechnology and in clinical market by providing user-friendly tools for researchers, clinicians, students to leverage this transformational opportunity of our lifetimes, because this is where the innovation is coming. This is where the latest technology is heading towards. Today, we will be learning about the overview of topics as promised that will be covered in the program. So let's start discussing about the basics of bioinformatics, big data, and these concepts and applications. So in bioinformatics, if we talk about big data, so the biological experiments produce multitude of data today. So compared to 20 years ago, so before that, we were using single experiments to generate data. Now there are thousands of experiments, automated experiments that are generating this multitude of data. And then we have open source databases like NCBI. Scientists from different part of the world can share their data and can collaborate with ease. So that's the new world. And we have now crossed the Excel barrier as well. So earlier we were used to use, I mean, most of the time we even use Excel, MS Excel for collaborating and sharing data. So bioinformatics researchers who are now are adapt with statistics, data, and programming on top of domain knowledge. There are also demands such as learning tools such as Panda, NumPy, Matplot, RShiny, all of that actually efficiently help us to maybe use machine learning as well. So this is right now is the market in bioinformatics. There's a huge opportunity associated with it also, because it's a growing market, and it has a lot of demand for all the biomedical research that is ongoing, disease related research, and many more applications that today we will discuss. So learning about this, I mean, what is the gap, where we are, I mean, what is lacking and what we can do. So breakthroughs in molecular biology, advances of precision medicine, biotechnology, and agriculture have translated into advanced technologies, right? Because now we are generating data with ease, and that simplified generation of accurate and in-depth data is is something like a blessing in a way, but at the same time it poses like a lot of questions, because there's a lack of analytical power and appropriate skills that creates a huge gap, because we have high quality data now, but we need experts to understand that data and make sense of that data. So those expertise and skill sets are lacking, and many of because of this, there's this gap between data and information in this field of computational biology and bioinformatics. And from a standpoint of what has been seen as, so bioinformaticians as such were viewed more like computer scientists before, who could manage a database, maybe run some algorithms. But modern bioinformaticians are different, and I'm going to tell you why. Because modern bioinformaticians have to identify what questions a researcher should ask, how to look for right answers. That's a very important part. And then what to do with this large amount of data produced from these studies. That is also a job of a bioinformatician to learn and understand. And also for all those people who are in biology and are now progressing towards learning more about biology and its application. So in bioinformatics now, as I was telling you about the opportunities, these scientists who are being able to do all of this, who can ask the research question, find data set, understand what answer that data set can give, these scientists are now integrated into every research team, making them into the core of innovation. So it's very important. And these So biology and as a field has been now ushering with the age of AI, big data with it, and creating a new line of scientists. That's where I'm coming at, which is bioinformaticians and computational biologists. So now about bioinformatics and the different kind of challenges. So when we talk about technical demand that prevents us from learning these skill sets because of the computational power that is sometimes required for us to do all this analysis. So in most of the cases, that means that our ability to analyze data and extract meaningful information is lagging behind because of the pace at which the new data is being generated. So oftentimes, like clinician, biologists, students from life science background report about challenges with data analysis and interpretation that is holding them back and limiting their ability to rely on advanced technologies such as genomic sequencing, which we could incorporate in our studies, in our research, and that could actually give us a really bright, clear picture of what is going on. So, if I talk about bioinformatics and computational biology, the first three words that Wikipedia used to describe bioinformatics are it is an interdisciplinary field. So, when we think about these three broad fields, for example, biology, statistics, computer science, or you can say instead of statistics, data science. But what exactly these disciplines are, and are there other fields within this disciplines? So when it comes to application in the context of biology, the current realm of molecular biology, the current realm of biology includes molecular biology, genetics, genomics, proteomics, metabolomics, systematic evolution, pharmacology, biomedicine, health sciences, and the list goes on. So this is not an exhaustive list, but when it comes to computer science, there are like obvious ones like software engineering, database management, information engineering, natural language processing, image processing. So these are some of the fields that are associated with the interdisciplinary nature of bioinformatics or computational biology, and how we can now being able to utilize these different areas of these different technologies into one area of biology. So what is bioinformatics? I think it's a convergence of biology and data where both are equally important. So we've been talking about data. So let's understand some of the data types, which are like some of the genetic data types, omics data types. So omics data includes phenomics, genomics, epigenomics, epigenomics technologies, and that can be used to. These technologies are so important in today's world because they can be used to explore the roles, relationships, actions of various types of molecules that makes up our cell, that makes up a cell of an organism. So many of this omics data can be generated using the NGS technology, which is commonly also known as high throughput sequencing. And this data shows, as I was telling you before, this data shows detailed information about genomic variants, pattern of gene isoform expression, and for an entire organism and for the entire time point. So the application of these technologies is something that is really exciting, and that we will be learning about in the next few slides. So one particularly exciting area of bioinformatics is its use in precision and personalized medicine. I think many of you have heard about it. So what is happening is doctors are learning more and more about the influences of the gene on a person's physiology, a person's metabolism, and more importantly, how drugs interact with their exact phenotypes. So these are some of the things. Other than that, using patients' personal genetic makeup and comparing it with the database of thousands of other patients, doctors can make more informed decisions about treatment and therapeutic choices. So these are some of the possibilities that are already happening in many places. Some of the drugs may be more efficient for a patient than others, while some of the drugs may have more unwanted side effects for some other patients. All these could be, theoretically can be evaluated now, given the hospital have I mean, maybe hospital or the organization have an access to those databases, and then they have the expertise to compare and find similar genotypes that could be treated with similar drugs. So there are different applications in medical sciences that could help make the entire healthcare system or the medical science more robust, empirical, and casual than correlational and uncertain, right? So most of such data types are generated randomly at a high speed. These sort of data sets are, generated at a really high speed, and these require like complex computational data processing system, a procedure to generate structured data sets which can be visualized and analyzed statistically. So that is some of the challenges that are associated with such high throughput data. But I mean, this is a program where we would want to simplify that and take you through this entire process of how we can actually do this by learning the logic. Now, this about this training program. So in this first training program, we will have live sessions every week. And the first live session will be about orientation and introduction to Omicslogic coursework and access to the multi-omics user-friendly TBioInfo platform. So after the program registration, and many of you might have already registered for the program, the first week, we will have participants and we will have orientation and introduction session. In this session, we will be collaboratively working with students to complete their academic profile on Learn Omicslogic and set up accounts on Big Data Analytical TBioInfo platform. So about this, I mean, anyone can join the Omicslogic Bioinformatics community for free, and that we will be showing you in a while. So in this week, we will also learn about big data application of bioinformatics. And after we do the sign up, which we will be doing in the next few slides, I'll be taking you through the hands-on courses and the premium courses associated with each week for you to learn all of this on your own. So in the next session, after the first orientation session and the introduction session, where we get to learn about you, about your interest, about the area that you would want to work on, maybe, if not about the basic training, the help that you need to go to the next level. So in the next session, we will be learning about this various NGS technologies, starting with genomics. We will learn to analyze NGS genomic data. And to give you an overview, genomics is an essential subfield of bioinformatics and is a major force in expanding human knowledge for genetically associated associations with disease and other traits. It's an interdisciplinary field again, and it includes development of methods for DNA sequencing as well as big data analysis for genomic sequences. So NGS data or NGS techniques allow whole genome sequencing and then analysis of epigenetic factors as epigenomics for DNA protein interaction, DNA methylation with, I mean, it is very efficient, and it has very efficient way to identify those. And there are massive databases. Obviously, I talked about NCBI, there's GeneBank, EMBL, SRA for you to do data-driven discovery and knowledge discovery. So this session serves as an introduction to the subdisciplinary field of genomics. Participants will get familiarized with biology of genetics, genetic variation, and other practical applications. And to tell you, I mean, I will be telling you more about this entire week once you all are signed up, so that we can have a demonstration of what will be covered. So in the next session, after learning about genomics, we will learn about metagenomics, or you will have an opportunity to learn about metagenomics. So this is a 1 month program, and I would want to clarify that you will have a lot of resources that you can complete in that period, or you can extend, keep on extending and keep on learning about different data types. So yeah, obviously there's an opportunity to do this right now in a month's time, but I mean, many of you would want to go and deep dive and can do these analysis after subscribing for the program every month. So in this session, we will learn about application of microbiome metagenomic analysis in life sciences. And as you know, metagenomics is to study a collection of genetic materials or genomes from a mixed community of organisms. Metagenomics is usually refers to study of microbial community. And we have also different projects in metagenomics to explain different sort of analysis that I'll take you through after the login. So after the metagenomics section, we will move to another section of analysis, which is transcriptomics analysis. This is like high throughput gene expression data analysis. So in this session, we will be doing the analysis using Omicslogic resources and the Bioinfo platform that we will be talking to you about very soon in the session. So transcriptomics, to give you an overview, is to study organisms' transcriptome. The transcriptome refers as a sum of an organism's RNA transcript, right? So the DNA information that is there in the genome gets converted into RNA through a process called transcription. A segment of DNA that gets transcribed into an RNA molecule is called the transcript unit. So studying and understanding the transcriptome gene expression levels help us understand that what are the proteins or what are the key genes that were maybe expressed differently in a different condition. So it is also about comparing these different expression profiles that we get from different experiments, and that technology and will be also covered, like doing differential gene expression. How would you do that? How can you get from raw data to a structured table of expression first of all, and identify which are the important genes that have shown this variation by using different type of statistical analysis. So we will be learning and we will also learn to use these various statistical methods of data analysis using supervised and unsupervised machine learning that can be applied to biomedical data or different kind of other data types. So using various examples of omics data, we will see how such analysis can yield significant results for various biomedical applications. And I quoted this because it's very important, because when you are looking in for information from a very big data, it's like finding needle from the haystack. So significant result is something that is very important and could be, if it is a research, it could be publishable as well. So about the Omicslogic program, in this Omicslogic beginners training program, the participants will have an opportunity to learn about various technologies, tools, algorithms, and pipelines, and workflows, and data sets, and research publications. And these are some of the 6 data types that you can see on this slide. So together with this existing modules for which you will gain access for the Omicslogic Learn portal, where you can explore these modules, exercise the pipeline building. How can you build a pipeline? How can you do coding from the very basic and apply it for bioinformatics, not hardcore programming, and various methods of analysis using project examples from peer-reviewed publication. But to start with this, I mean, we will have to do the login or the sign up together. So now let me invite Sonalika again, our community manager, to help us create a login, show us all how to create a login, and then we will start exploring these resources that I just discussed.
Thank you so much, Dr. Mohit. Now, I would request you all to go to our portal, that is learn.omicslogic. I'm sharing the link with you all in the chat box. It would be great if you could follow along. Now, once you are on the portal, you need to log in. If you have not created an account yet, then you need to sign up. Those who have signed up already, please let me know in the chat box by sending a yes or one maybe. And those who have not yet signed up, you can go along. Here is the link for you all in the chat box. And you have two options to sign up. It would be great if you could create an account for yourself with your mail ID and create a password as well. Or you can also log in with your social account. Let me know in the chat box if you're going along. Thank you. What about others? Have you logged in? Let's wait for 5 minutes maybe, or 2 to 3 minutes for you all to log in. Please let me know in the chat box. I'm waiting for you all to log in. Thank you, Priyanka. Thank you, Ruby. Thank you, Sonia. For those who have logged in, let me just tell you how you have to begin with. So to begin with, first of all, you have to update your profile picture and also link in your social account and ensure that you have your name in the correct format. It should be title cased and not in capitals or not in small, because this is the name that would be reflected on your certificates. Yes, Madhumita, I can wait for you. Thank you, Chika. Those who have already updated their profile, it's good. Others can update their profiles in between the session or later as well. And you can see all your activity in the profile tab. So let's say you started with a course on genomics, or let's say bytes and molecules. You can see how many lessons you have covered and how many are still to go. And once you have covered or completed all the sessions, you would be able to get the certificate in the certificates tab in your profile section itself. And after joining the program on the Omicslogic Bioinformatics and Computational Biology, you would be able to see the program under your profile. And as the program has not started yet, that's why it shows 0 out of 4. So once the program would begin, I would be posting in all the recordings for the program on the program page, and you would have to complete the sessions to get the certificate. On the program page, we have mentioned about what all would be included in the program. So we have 4 workshops in total, and Dr. Mohit would be explaining about each and every workshop in detail. And I would be uploading the recording of each and every workshop once it's over. And also you would be able to find the schedule for the program. So I had a message in the chat box where the participant was asking about the schedule. So we have 4 sessions scheduled on 2nd August, 5th August, 9th August, and 12th August. And also you would be guided towards the asynchronous course material. Have you all signed up? Please let me know in the chat box. Sorry. While others are signing up, I would just want to quickly launch a poll to know about you all in a more better way. My first question is, what is your educational background? Are you an undergraduate, graduate, postgraduate, PhD, or a postdoc? And my second question is, what is your current position? Are you a student, researcher, faculty, or are you from management and administration? And my third question is, what are your research interests? Is it infectious diseases, genomics, transcriptomics, metagenomics, data science, machine learning, or bioinformatics using R or Python? The poll would be up for a minute. Thank you for giving out your votes? I can see 40 people have already voted. I would request others to vote as well. Yog, you have to go to the profile section and update your profile after you are done with signing up on the portal, and we will be sharing the program page link with you all for you to register.
Thank you, Sonalika. If I may take over now.
Yes, sir. Over to you, sir. Thank you. I'm
Okay,
ending the
so
poll. I hope everyone has voted. Over to you, Dr. Mohit.
okay, thank you, Sonalika. And if you can see my screen.
Yes, sir, we can see your screen.
Sorry. So let us go to the Omicslogic Learn, and I've logged in. I have a profile here, as Sonalika showed all of us, that you can create your profile and you can have a picture, and you can have your name and social media links if you like. You will see the courses that you complete and the achievements that you did. You will also accumulate points for your achievements. So I've accumulated almost 5,000 points and I've gained 2 certificates so far. So you can keep on doing the courses and gain a lot of certifications for the courses itself. And for program, if you're joining the program, you will also be eligible to get a program certificate. Now, going back to some of the questions, whether this program is a paid program, how do you access the resources that we discussed, how to go about the training, and what are the next steps? These are something that I'm going to tell you about in much more detail now. So in Omicslogic community, as I said, it's a free community. Anyone can join for free. Here you can see register for free. But there are premium courses which requires you to utilize the T-Band server. So there's a subscription for the program, a very minimal cost that we have kept together. And that's why we are calling it a special opportunity for you to learn from us and learn from the resources that are available. So you can join the program to access the premium courses. Otherwise, you will have access to the free courses. I will tell you which free courses and how you can access those as well once you have created a login. I'll do that. But before that, I mean, if you are interested in going through the entire, sessions, going through the workshops by experts, going through the premium courses and all of that, then you have to join the program through this page. And it's kind of simple. So let me show you. I'm already a part of the program. So if I have to join the program, how can I join? Yeah, obviously you'll have to log in from your account first, and then you will see the program page. And once you go in this program page, or if you can also like go to the link directly, you will be able to come to this program page, and then you will have to complete your registration. So you can give your information, and we will be sending you more information about the program, more details if you are not sure about what will be covered. And at the same time, you can start your application and you book your seat. So it's a program that will be available for anyone to take. And you can join this entire, whatever we covered in today's session will be available with the beginner's license. So we have different subscription levels. So the first beginner subscription level is just for $10 a month, which will give you access to all of the things that we discussed today. And we have different other subscription levels for you to do research, for you to do work on project, and maybe we will discuss it in the next time when we meet. But for this program, you will be able to do all the coursework and all the live sessions that we just discussed. And about the coursework, I'm going to tell you what all coursework are there, which are for free and which are for premium, and why they're premium and why there's a cost. So let me go back to the slide deck. Yeah, the first course that we have put together for everyone to take is a free course, other than the ones that you see in Star or the premium ones. So when you do this course and go through this course, Bytes and Molecule, you get an overview of a very basic understanding about the different data types that we will be discussing in the program. So anyone coming from a background without knowledge of biology can start taking this course, Bytes and Molecules. So let me just quickly show you a video for the same.
Bytes and molecules. Every living cell has its own hard drive where it stores bytes of information needed to function and grow, adapt to environmental changes, and fight off dangerous intruders. This information is stored in molecules that are arranged in sequences of letters, referred to as DNA code. In the 1990s, a tremendous effort by governments, researchers, and companies resulted in the full sequence of the human genome. Since then, we've seen an exponential growth of molecular data that describes cells, tissue, organs, and organisms. Massive data sets containing genetic information about the young, old, healthy, and sick are now freely available to the public. Here, we will explore the various omics data types as well as examples of the information they contain, genomic variants associated with ancestry, gene expression levels responsible for cancer spread, and protein modifications that can render important code unreadable. This course is intended for all those interested in understanding how the current revolution in biomedical data impacts biomedical research and human health.
So as you learn from this is the first course that you will go through. And it's a free course anyone can take. And in this course, you will learn about the different data types. Another free course that we have is the Introduction to Bioinformatics. It's a course that takes you through different types of application of bioinformatics, big data bioinformatics, as I said. And it's a course where we get to learn about you, your expectations, what do you want to learn in bioinformatics, which area of research that you are interested in. So this course has a form. When you start taking this course, you will get a form, and I'll request to Sonalika to share maybe the link to this course as well. So this is an introductory course where you will get the overview of different type of uh, mm, uh, fields where you can apply bioinformatics, and you can also tell us about your area of research where you are interested in. So these two courses is something that we would want you to start with, even if you are joining the program, even if you're not joining the program, it will be really helpful for you to go through these courses. So once you go through these courses, we also have other free courses, introductory courses are for omics bioinformatics. As I said, that in this program you will also have opportunity to learn coding. You will have also have an opportunity to learn this from the browser itself and get points and recognition and all of that by while completing this coursework. So in this first tutorial, which is just to get started with bioinformatics, it tells you about loading structural data into R, a multidimensional data visualization in R, reading a very basic file, DNA file, and then understanding the DNA replication and reverse complements in R. So this is here also you will have free courses where you can learn about these different technologies, and how it can be applied, different programming languages, how it can be applied for data visualization. Now, I mean, I told you about the premium courses as well. So the premium courses, how they can help you do the hands-on analysis on yourself, by yourself, at your own pace. So let's take a look at that. Let's have a demonstration about that as well. So let's go to the Learn portal again. And now what we will do is that we will go to the section called Courses, and we will find all the courses that we are interested in. If you are interested in Python, we can try Python. If you are interested in R, we can do R. If you're want to learn about the introduction to bioinformatics and genomics and then epigenomics, then all of this course material is here. We will just have to search for them, and and I'll show you that's pretty simple. Okay, I'll have to log in first. So I'm logged in now. I can see the courses. So let's take a look at the R course. So we have different courses, as I said, getting started with R. Another course is Introduction to Data Science using R. Now we have a lot of lessons within these courses that you can take and earn points from. So let me show you how it works and how it is so simple and easy to do this, that anyone can learn from this. Because when you start from basic, you tend to get an understanding of what's going on, and you understand different type of commands that are being used and get familiarized with it. So we start with you can install R locally on your computer as well and try this out, and you can do this from the browser itself as well. So with every tutorial that you see here, the first part is about is to understand the problem, what the problem is all about, what we are doing here. So we are looking at different type of data, categorical data or continuous data in data science, and then we are trying to do some plotting to see the characteristics of data. I mean, how they are placed, if we can separate them or not. So this is being used in many of the bioinformatics application, especially when we are doing big data analysis. So here, I mean, you will learn to upload the data, how you can use links and upload that data into the browser, and then you can run different sort of commands to analyze that data. So once we learn about these different commands, which is just shown here, you can go through them and easily learn about them. Then there is a breakdown of code that will be used for this tutorial. So what this code is doing, so it actually explains all of it and what kind of results that we can expect, and what kind of analysis that we can do from this sort of data. So once you do this, you get to a point that where you can practice it yourself. So it's a code playground where you can write your own code, and you can load data and you can run the analysis here itself to show that if you're getting the result or not. As you see, as you can see that this is already a code that is put together and is working. You can change this, and you can run your own code and do your own analysis, downstream analysis, using this code blocks and different tutorials that have been shown here. Uh, that have been shown here in the Learn portal that you will have access to. So the opportunity is, there is a lot of opportunity to learn about different things. So I just told you about this R code. Now let me go back to the slides, and I'll show you what all different courses that we have put together for you to take. So you can start from Python as well, similar courses. You can apply and see what is the difference, how you can learn both of them, and I mean, choose what you are, I mean, more comfortable in, and what kind of analysis that you are into, and what kind of research that requires to use these languages and apply these languages for data analysis. So another thing, another important aspect of this sort of learning, Omicslogic bioinformatics training, is to be able to access the T-Bioinfo platform and run your own analysis and get the structured data to analyze further in R and Python. So when we go through these courses, such as genomics, as I said in the second session, after the introduction session, we will be doing genomics. So in genomics, we will be going through very basic, starts from the DNA structure and variants, why we are using the genomics technology, and where it has been helpful, how we can identify variants and identify mutations that are critical. So, learning about that, working using R, then introduction to genomics, where you get an overview of all of that in much more detail and depth. and then practicing using the pipelines. As I was telling you, that you will be getting an overview and understanding of how algorithms are connected together, and they give us a structured result after doing a complete analysis. So there are several tutorials, and the ones that are starred uh, includes practical assignments. So for that, you will be able to access the tBioInfo server to run some example pipelines. And now let me take you to the tBioInfo server and show you how it works and why it is so easy for anyone to use. I mean, even students without a background in bioinformatics and computational, computational sciences can do this. So this is the tBioInfo server, and once you register, we will be sharing the access for you to take some of the example pipelines from here. And now let me show you an example pipeline from the Transcriptomics data analysis. So it's an AI guided platform, and we have put together examples from publications that you can take and learn from. And at the same time, you learn about the logic. So many of these algorithms that you see here are open source algorithms. So our purpose is not to make you dependent on a platform for data analysis, but make it simple so that you understand how the algorithms are used, what are the outcomes, what are the outputs that you get from this sort of analysis, and how to interpret that result. So understand the logic behind the analysis is what we are focused on. That's why we are called Omicslogic. And to do this, I mean, we have put together many examples for you to go through. One of the examples that I'm taking you through is the cell line data set. So it's an RNA-seq experiment where the scientists or the researchers have generated data on cell line using cell line, breast cancer cell line samples. And then we are processing the raw data here using these pipelines. So we have already put together different sections for different sort of analysis. For example, pre-processing, mapping, quantification. These are different sections that you have to go through to process data and get a structured table. And now one thing that you can see is that they are differently colored. So to make it more easy and simple, they are color coded so that you understand that these sections are used for pre-processing, these sections are used for mapping, this is for quantification of expression level. And what are all of these steps? And we will be going through in detail in the workshop session. And also they are described, I mean, absolutely amazingly in the courses that you will have access and you'll have the opportunity to go through in this training. So the input file and the output file are already stored because it's an example pipeline for you to learn. So the first step that we do in such analysis is to clean the data, make it more structured. So we use algorithms such as Demomatic, PCR clean, and then I'm going to show you how we can map it to the transcriptome and then generate a table of expression. So if you can see, I should be a little slow, I know, but it's a quick demonstration and we'll be going through this in much more detail in the session, and it's described in the courses as well. But here also you will be able to learn about these pipelines, these algorithms, what they do, what paper that you can read about them also and learn about them in much more detail. So here what we did is that we pre-processed the data, we mapped it to a transcriptome. And when we say if we are mapping or aligning it to a transcriptome, we are doing it on a reference transcriptome. And then we are generating a table of expression after doing the alignment or the comparison. And then we are ending the pipeline. And here you will see that there are different courses that you can take and go through, which are on the Learn portal. Now, during the session, we will be able to tell you the output and how to analyze these results and what is the outcome and what can you do next. So the next step is to take this data, structure table, run some other analysis and do some machine learning analysis based on the research that we are going through. So this is the way we kind of go and explore the tBioInfo platform. And obviously it has also the capability to do the analysis right from NCBI. So you can upload data from NCBI, you can upload data from your own research lab, and those are available with different subscription options to access this platform. With the program, you will be able to use it for educational purpose. You'll be able to do example pipelines and learn first before you do your own experiments. So that's the process that we follow for you to go through the technology. First, go through the steps, do the data analysis on example projects, and then learn on your own or public data that is available, and that we discussed that it's so huge that we have been able to do a lot of quality research out of that data. So this is about the TBioInfo platform that you will have access to, and this is the coursework for genomics, and then we have the coursework for metagenomics. It's very elaborated coursework. As I was telling you, there are examples here that you can learn from, and learn about the OTUs, learn about different types of metagenomic analysis, learn about the data to pipeline, phyloseq. All of this is covered in different tutorials under this section. So if you go to the course for metagenomics, you will find all of them bundled together that you can take one by one and learn about and apply this to your own research once you've been able to go through this material, which is mostly hands-on and also gives an overview of all the technology. And there are also videos in the tutorials, so you will be able to go through the videos, recorded videos, and replicate that using the TBioInfo platform, because at the same time you will have an access to do this hands-on thing. So that's why this Omicslogic training is a really unique opportunity to learn for real. So the next technology that we will be covering is the transcriptomics technology, and we have detailed explanation of this transcriptomics data analysis. We've been doing several programs and have a lot of participants who have gone through this training and come up with unique projects that have been published, as well as got recognition in various conferences and presentations. So another aspect of doing an advanced level analysis is to learn about the application of machine learning, and we use different type of machine learning regression. So there are factor regression analysis, supervised machine learning, unsupervised machine learning, to kind of get insights and results from transcriptomics data analysis. Another advanced section that is again available, I mean, as I said, it's a subscription-based model. If you are into this, then you can go in deep into this and maybe, try different courses, run some ideas that share with us some research ideas that we can work on. So there are different ways we can collaborate and do research together. So single cell transcriptomics is such an area, very promising area. A lot of modern research and high value research is coming out of, high impact research is coming out of single cell analysis. And there's a lot of data that has been deposited as well. So another aspect of doing this, all of this, is to learn machine learning, how we can apply machine learning BioML. That is also is in there in the coursework. So you will also have opportunity. I know that there is a lot of coursework that you can do and learn from. So you can also take your time and go through this analysis at your own pace. And at the same time, learn from the video that will be recorded and provided to you. And we will have also mentors that you can, connect with and learn about specific sort of analysis. All those opportunities are available on with us for this Omicslogic training. So that is about the coursework that is there. Other than that, I have already told you about the data science part for R and Python. And then we have mentors and then we have programs, and which I can show you actually quickly here on the website. So when you're logged in, you have option to do coursework, you have option to select a program, and you have option to do research. So we believe that it's best to start with the coursework, go through the examples, and then conduct your own research and learn from specialization programs. One of such program is this one, that is Omicslogic, Bioinformatics and Computational Biology. People interested in infectious diseases, data science, transcriptomics, we have specialization programs that we keep on doing as well for those. But to those who have just started, this is a best opportunity for you to take Omicslogic, Bioinformatics and Computational Biology. So that was about my presentation, and I hope that you were able to understand what I have covered and what we have covered and talked today. And if there are any questions, I'll be happy to take those questions. And at the same time, it would be great for you to ask them right now. And then I would like to maybe invite our CEO, Ilya Brodsky, to tell you a little bit more about research and the opportunities with research that we have. We have a symposium coming up on August 21st and 22nd, where we will be showcasing all of this research that our interns are doing, and then there will be opportunities to talk about your own research as well. So over to you, Ilya.
Yes, thank you very much, Mohit, and welcome everyone. Great to see all of you here. I will just briefly introduce the symposium, just kind of starting off with why this is the right time. So as Mohit has already mentioned, there have been numerous breakthroughs in molecular biology, including advances of precision medicine, biotechnology, agriculture. All of these have translated into advanced technologies that actually simplify generation of accurate and in-depth data. But the lack of analytical power and appropriate skills creates a huge gap between data and the information that is needed in many of these fields. So there's actually a gap between the lab and people who make decisions. And so to bridge this gap, we started looking into some information from people who are involved in this kind of in these kinds of tasks. And the survey clearly shows that the majority of users that are attempting to run pipelines and analyze large-scale, high-throughput omics data are struggling with the data analysis component. And so we understood that there has to be a solution that is designed for early on, while there are students in high schools, colleges, and universities, to actually supplement their educational curriculum that they already have, introducing them to these challenges early on and building a pipeline for them to develop research skills and practices in applying bioinformatics analysis tools to large-scale data sets, and actually developing research projects to showcase what they have learned. So not a skill-based and a certificate-based course, but a research experience. And so to introduce how this could be done and discuss some of the challenges that exist in this field of bioinformatics and data science for life science data, we are going to hold this Omics Research Symposium, which is going to be dedicated to the future of bioinformatics education and will cover critical challenges, and we hope to identify solutions for improved educational strategies that are focused on biology as a data science. And we have a great panel of speakers that are experts who will be addressing the pressing challenges with data science education for all life sciences students and explore how to best equip all life science students in data skills for the 21st century. Our speakers will bring their perspective as researchers working on cutting-edge topics in bioinformatics. They are directors of successful degree programs and who are seeing their students continue with successful careers in education, academic research, and industry. And together, they will kind of provide us with a comprehensive view of both the opportunities for students and the challenges to be addressed, and hopefully make the most out of their studies and focus on acquiring the skills that matter and that will really help them become successful and make a true difference in these emerging fields of biotechnology, precision medicine, bioinformatics, data science, and others. So what we will discuss in the symposium is, number one, for students, how do you stand out from the crowd when you apply for academic programs or careers in bioinformatics? Number two, for universities, how can universities equip their students with relevant skills and experiences, providing them with an opportunity to stand out? And then we will also discuss the future of this field of bioinformatics as a whole, how it's changing. It's changed dramatically over the past 10 years at least, and where we can find these cutting edge challenges that someone can contribute to. And these include, you can probably think of them yourself, CRISPR technologies, right? Revolutionized genomic engineering. We have genomic sequencing that is under $1,000 for the whole genome. So the sequencing technologies have been changed dramatically. We can also see the application of machine learning and AI, DeepFold, recently released, right? So we all hear about that. And obviously mRNA vaccines. So the translation from research to application has been tremendously sped up already. Drug discovery, drug repurposing, all of these challenges have been undergoing a dramatic change. And so to participate in this revolution utilizing data science, what our interest and our goal is to identify effective practices that allows students to participate in these challenges. And the way that they can do it is by learning first about the challenges, understanding the tools, just like you are listening today to this program, and then applying them. And applying them means developing research projects. And so here we will also showcase student projects that have been completed over this summer and the past several months. We have hundreds of students working on these kinds of research projects, and through the same portal that you've already registered in, you will be able to see how this happens. Those will be evaluated by our team of mentors and judges with expertise in bioinformatics, medicine, microbiology, biotechnology, and others. We will also highlight some of the exciting updates to the T-Bioinfo platform. The platform covers many different types of bioinformatics applications, but it also includes some new sections that are going to be presented by the team at the Tauber Bioinformatics Research Center. And those include technologies for single-cell genomics and multi-omics integration, advanced mathematical modeling for different processes related to the field of virology, ultra deep sequencing technologies, and the integration of multi-omics data. We will also share some exciting updates about the Omicslogic training modules for biology as a data science. And we will discuss some of the regional collaborations that we have in the United States, India, and Africa, highlighting important modules that can be applied to oncology, infectious diseases, neuroscience, and agriculture. We will introduce new modules and project examples for precision medicine as a preclinical and clinical training option. We will discuss how this pandemic gave public awareness about infectious diseases and what kinds of hands-on exercises students can access to learn about the viral, bacterial, and parasite-caused infections using largely available public domain data. We will discuss advances in agriculture and the rise of technology-driven precision agrotechnology with a focus on sustainable food supply, climate-resistant strain breeding, and methods of bioinformatics analysis that are in high demand in this industry and what areas of research are in pressing needs. Finally, we will introduce our new and planned specializations like the field of astrobiology that is captivating the minds of students who are seeing the commercial space industry take off and develop before their eyes. So together with our existing modules for which you will gain access, so you already have registered and you will have access to the basic modules, you will explore them, including all the different exercises that have been already mentioned. So importantly, what we will offer for students who are joining is also some exciting opportunities to join academic programs. So if you are considering education in bioinformatics, a formal education in bioinformatics, there are many different universities that offer such programs, but they're not the same in every single program. So every program has a perspective and a focus and a special variation to the curriculum and the objectives. And so we will bring directors of these programs to how their program is different, what is the program focused on and specialized in, and how a student can join. And we will have presenters from Carnegie Mellon University, Louisiana State University Health Sciences Center in New Orleans, Georgetown University Systems Medicine Program, Omics University, University of Ghana, and others. And the objective here is really to offer opportunities for students who are entering this field or who are already in this field, from data generation, from bioinformatics analysis and integration, and all the way to decision making, to really provide them with a comprehensive bioinformatics training in biology as a data science. So you can register for free just to attend. If someone could please post the link in the chat box on how to register. If you register and you have a project that you would like to present to our judges and evaluators, you can also submit a poster and that poster could be either just presented or it could be showcased if it wins a prize. We have cash prizes that best posters will receive in the different categories that we have. And importantly, you will get recognition from many different respected universities and research centers that are going to be there and represented. We will also have companies. Those companies will be talking a little bit about how they are providing opportunities for people who are starting their careers, students and recent graduates, to get into internships and various opportunities to start their career with biotechnology, utilizing bioinformatics and data science. So I'm excited about this. Again, this is August 21 to 22. Thank you, Mohit, for sharing the link. So the link is in the chat. You will see here that you can simply submit this form to participate and learn a little bit more about the different people who are involved. What are the different topics? You can submit your research poster if you're interested, and also look a little bit into what else we are going to be doing. And here at the bottom, you will find a video presentation. And of course, we are all excited to join this, and not only to learn about these pressing challenges, but we also want to get to know all of you, the community that we interact with regularly, to learn more about what you are doing. And we also have some hiring ongoing. So we're hiring students and researchers as well. So we are looking to connect with you there as well. Thank you, everyone. That's all I had to share. So back over to you, Mohit or Sonalika.
Thank you, Ilya. I think if there are any questions, we can answer those, and then we can conclude the session, Sonalika.
Maybe we could just also mention our social media groups where people can join, and we can learn about them more there as well.
Yes, so let me quickly share that with you. Thank you, Sonalika, for posting the channel link. So you can join our LinkedIn channels where we keep on posting about the job opportunities. And you can also learn about what is the research which is going on and what are the programs that we are doing and the events that we keep on posting about by joining these groups. You can also join us on Facebook using the link here. 1 second, please. Sonalika, if you can also share this Facebook link.
Sure, sir. I would be doing that just in a second. So being a part of our LinkedIn group will also help you get in touch with a lot of researchers and know about what's going on around, and also stay updated with all our programs and courses. I'm sharing the link to our Facebook page. And also we have a Telegram group and a WhatsApp group so that you can stay in touch with us, and also a Slack channel for better communication. I can share the links as well. And also if you have any queries regarding the program, I would be sharing the program registration link once again with you all. And if you have any queries, you can mail me at marketing@pine.bio or support@pine.bio. Let me just quickly put it at the chat box.